Welcome to our presentation on oncolytic virotherapy. It dates back to the early 20th century when doctors noticed that some cancer patients improved after contracting viral infections. This innovative approach leverages the natural ability of viruses to infect and kill tumor cells, offering a beacon of hope for patients battling cancer. Our journey through this domain uncovers the mechanisms of action of oncolytic viruses, explores representative examples, discusses strategic modifications and combinations with other therapies, addresses challenges, and reviews the clinical status of this cutting-edge treatment. Oncolytic viruses act through a combination of direct and indirect mechanisms. Oncolytic viruses selectively infect, replicate within, and lyse tumor cells whilst leaving healthy cells intact. Upon infection of tumor cells, oncolytic viruses replicate and lyse the tumor cell, resulting in tumor cell death and release of viral progeny. This tumor cell lysis results in destruction of the local tumor microenvironment and induction of an anti-tumor immune response through local immune infiltration and release of tumor antigens. Immunomodulatory effects are significant. Some oncolytic viruses are engineered to express cytokines or other immune-stimulating factors, enhancing the immune system's ability to recognize and destroy tumor cells. Oncolytic viruses can convert cold tumors into hot ones through a process that involves selective infection and replication within tumor cells, leading to immunogenic cell death. This results in the release of tumor antigens, danger-associated molecular patterns, and pathogen-associated molecular patterns, which activate the innate immune system. The released signals stimulate dendritic cells, macrophages, and natural killer cells to secrete pro-inflammatory cytokines, enhancing T-cell priming and infiltration into the tumor. Oncolytic viruses also help overcome physical and immunosuppressive barriers within the tumor microenvironment, promoting a shift towards a pro-inflammatory state conducive to effective anti-tumor immunity. This process not only targets the primary tumor but also elicits a systemic immune response capable of targeting distant metastases, thus converting immunologically cold tumors into hot ones, making them more susceptible to further immunotherapeutic interventions. The diversity of oncolytic viruses is remarkable, with each processing unique characteristics ideal for targeting different cancers. Herpes simplex virus stands out for its ability to be reprogrammed into a less pathogenic form while retaining its oncolytic properties. Studies have demonstrated its effectiveness, particularly in brain tumors, where it selectively targets cancer cells while sparing healthy neural tissue. Adenoviruses are attractive due to their relatively mild human infections. They are engineered to replicate in specific tumor conditions, like as hypoxia or specific oncogene activations. The vaccinia virus, well recognized for its role in smallpox vaccination, exhibits strong oncolytic properties and an impressive safety profile, making it a candidate for various cancers, including melanoma. Reovirus, a naturally occurring virus, preferentially targets cells with dysfunctional RAS signaling, common in many cancers. Beyond these, Newcastle disease virus and Seneca Valley virus are emerging as potential candidates, each with distinct mechanisms that make them suitable for targeting specific types of tumors. Genetic modifications enhance oncolytic viruses in multiple ways. By altering their replication machinery, these viruses can be engineered to only replicate in cells with specific tumor markers, thus avoiding healthy cells. This is achieved by inserting or deleting genes that control viral replication. Safety is paramount, and to this end, viruses are engineered to be less pathogenic and more specific to cancer cells. Genetic modifications can also involve the addition of suicide genes, which can be activated to destroy the virus if necessary. Another innovative approach is to arm the virus with genes that boost the immune system, turning the virus into not just a direct cancer-fighting agent and also an immunotherapy tool. These modifications can be designed to work in synergy with the tumor's microenvironment, exploiting its unique conditions for enhanced therapeutic effect. In the quest for a more comprehensive cancer treatment, oncolytic viruses are being combined with other therapeutic modalities, such as chemotherapy, radiotherapy, and immunotherapy. When paired with immune checkpoint inhibitors, such as PD-1 or CTLA-4 inhibitors, oncolytic viruses can overcome some of the cancer immune escape strategies. This combination has been shown to be more effective than either treatment alone in some studies. Targeted therapies, which focus on specific molecular targets within cancer cells, can be directly delivered to the tumor site by oncolytic viruses, increasing their concentration at the target site and reducing systemic side effects. Chemotherapy, traditionally known for its broad action and side effects, can be used in lower doses when combined with oncolytic viruses, potentially reducing toxicity. This synergy can lead to a more effective and patient-friendly treatment plan. Furthermore, the combination with radiation therapy is being explored, with the potential for oncolytic viruses to sensitize tumors to radiation, thereby enhancing its efficacy. Despite the promise of oncolytic virotherapy, the journey is fraught with challenges. These include tumor bulk penetration, antiviral immune responses, off-target infection, adverse conditions in the tumor microenvironment, and the lack of specific predictive and therapeutic biomarkers. The solutions proposed include using junction openers and extracellular matrix modulators to improve penetration, capsid modifications, and bispecific adapters for better targeting, stealthing techniques and cellular carriers to manage immune responses, utilizing hypoxia response element-containing promoters, and identifying reliable biomarkers for patient selection. These solutions aim to improve the efficacy and safety of oncolytic viruses in cancer therapy. As of the most recent update, 4 oncolytic virus products have received marketing approval: Rigver in Latvia for melanoma, Oncarine in China for head and neck or esophageal squamous cell cancer, Imligic in the United States for unresectable cutaneous, subcutaneous, and nodal lesions in melanoma patients post-surgery, and Delatact for residual or recurrent glioblastoma, noted for its good safety profile and lack of transgene payloads. A number of clinical trials and translational studies involving oncolytic viruses have been filed at clinicaltrials.gov, with the most focusing oncolytic adenoviruses, HSV, and vaccinia virus. Oncolytic virotherapy has functioned well across different cancers, routes of administration, and both monotherapy and combination therapies. Apart from the traditional therapeutic approaches such as chemoradiotherapy, the recent more efficient combination therapies mentioned above are also being tested in patients with different cancers. Among these, immune checkpoint inhibitors and cellular therapies are increasingly being utilized in clinical trials with or without OVs, indicating enormous potential for combination therapies, especially in patients with refractory tumors. Taking advantage of the Oncovirotherapy platform, Creative Biolabs provides customized, standardized, reliable, and high-quality oncolytic virus therapy development services for clients globally. Our service covers all aspects from virus engineering, cell biology, to animal testing with a special emphasis on virus engineering and in vitro and in vivo validation. Various types of engineering systems have been established in-house to facilitate the oncolytic virus development with less time and reduced budget. GMP-compliant manufacture of oncolytic virus is also included in our services. Creative Biolabs provides a series of pre-made, purified reporter encoding and cytokine-expressing oncolytic virus, including herpes simplex virus and vaccinia virus. Except for the existing products, we provide customized, standardized, and reliable services to fulfill our clients' individual needs for various cytokine genes. Please feel free to contact us for additional information and a detailed quote.